All right. Thank you, everyone, for having me here. My name is Sumantra. I am a new faculty in the IISc Physics Department. And today I'm going to talk to you about how protein nanoclusters and dynamics is affected by the actin cytoskeleton. So because most of you do not know me, this is my fledgling group. So I have a postdoc, a couple of undergrads, and two PhD students right now who are all working on different aspects of biophysics of actin cytoskeleton and membrane biology. But today I'm going to talk to you about protein nanoclusters. So let me give you a brief resume of what nanoclusters are. So as the name suggests, they are nanoscale organization of different membrane proteins. For example, one of the most famous clustering protein is KRAS, which is also involved in many of the cancers. At least 1/3 of all known cancers are caused by RAS. And on the left-hand side, I'm showing you an electron micrograph of these proteins. So these black dots are representations of where these proteins are. On the right-hand side, there is a processed image. So when you do look at this processed image, you can see that there are many different colors. They kind of signify what are the different sizes of these protein nanoclusters. You can see that it can be quite heterogeneous. Sometimes it can have 6 to 8 proteins. Sometimes it can have only 2 or even monomers. So the interesting thing is not that they are static. The fact that they can be dynamic and transient and they can have a lifetime, that is what is important because that's how they manipulate the signals that come in and go out of the cell. So that's why they have been very important in understanding cellular signaling processes. Um, you can get some idea of the static features of these nanoclusters by defining this function called the Ripley's H function. I'm not going to go into the detail of what this is, but this is basically similar to the radial distribution function, G of R. And what is most important is the fact that there is a peak when you have clusters. The height of this peak basically tells you what is the intensity of clustering. The taller this peak, the more clusters you have. And the length at which this peak appears is roughly the size of these clusters. So this is what you should keep in mind. And this becomes important because now you want to basically probe what are the different biophysical mechanisms that control these clusters and their propensities. And one of the first things that people did was trying to understand how actin cytoskeleton is involved. And they used this drug called latrancolin, which disrupts the actin cytoskeleton. So when you add this drug, even at a small amount, you immediately see that the peak height goes down, which implies that the clustering ability of these proteins is actually going down. And as you increase, you see a saturating effect that which basically implies that there is some kind of dependence on the actin cytoskeleton. And that gave rise to this whole paradigm of actin-dependent protein nanoclusters. And this is what is the subject of my topic today. So our foray into this subject was kind of serendipitous. My friend Debanjan, whom some of you may know, so he was looking at RAS and RAF and their interaction on the membrane because he was interested in understanding how they cause cancer. So RAS can have RAS has oncogenic mutation, RAF has oncogenic mutation, and his hypothesis was that the oncogenic mutation tend to stay longer in the membrane, which allows it to interact more with these other proteins, ultimately giving rise to uncontrolled cell growth. Interestingly, we didn't really find any such difference. So in the orange, I'm showing you the normal variant, and in the blue, I'm showing you the oncogenic variant. As you can see, the lifetime distribution or the residence time distribution of KRAS kind of falls right on top of each other. What is interesting, though, is that this is not an exponential distribution. There is an exponential tail, but at short times, you start seeing power law distribution, so which is quite interesting. When you look at the residence time distribution of RAF, it is even more interesting. You start seeing this kind of power law at the short times, but at longer times, depending on the situation, you either get an exponential or you get this kind of a bumpy distribution. But this is a complicated story, so I'm not gonna tell you how RAF distribution got its bump. Today, my focus is more on understanding how the nanocluster lifetime whether nanocluster lifetime and the actin dependence can explain this power law that we are getting over here. To do so, we resort to one of the classical theories of actin-dependent protein nanocluster that was proposed by Madan and his group at NCBS. And what they suggested was that this actin cytoskeleton, because of its dynamics and interaction with the myosin motors and whatnot, it forms this transient structure called actin esters. And these proteins, which are membrane proteins, they can bind to this one of the actin filaments and gets advected towards the core of this aster, forming this kind of a circular shaped nanoclusters. Okay. So we took this model literally and said that, okay, let's assume that we do not want to worry about the actin-aster dynamics. For us, there is always going to be an aster. All we are going to worry about is the fact that the proteins can adsorb and desorb with some rate, k on and k off, and they can form this protein nanocluster. Okay? And then this protein nanocluster can decay through either of the two means. They can decay either through aster fragmentation, which is going to happen with some time scale Ta and tau A, and then the proteins can just simply desorb because it is a stochastic process, which is happening with some time scale tau D. And the lifetime of this protein nanocluster will be then given by the minimum of these two time scales. Okay? So how do we then go about modeling this system? And we made a really blasphemous assumption that everything is well mixed, and we're going to use some kind of chemical master equation and solve it using the Gillespie algorithm to try to understand what is going on over there. So what we do is we say that, okay, the protein is going to bind and get advected, and that is basically going to increase the size of this cluster by 1. But because of this stochastic binding unbinding process, it will eventually grow up to a maximum size and then decay back to the size 0. So this entire time is going to be our desorption time, and the maximum size is going to be the cluster size. Okay? And then we're going to assume this binding rate is going to be proportional to the concentration of actin at aster. And for simplicity, for now, we're going to assume that all asters are identical, so the concentration C is identical for all asters. And when we run our simulation for various k on and k off, we get very, very different distribution. So in the red, I'm showing you the cluster lifetime distribution. In this sort of dotted line, I'm showing you the aster fragmentation time distribution. And in this long dashed line, I'm showing you the desorption time distribution. And what you find interesting is that sometimes this cluster lifetime kind of falls right on top of the aster fragmentation time distribution, and sometimes it falls right on top of the desorption time distribution. You can make it more quantitative and sort of make this phase diagram of sorts, which shows you that depending on this duty ratio, which is a function of k on and k off, you get fragmentation dominated in this narrow corner, but most of the time you tend to get desorption dominated. What is interesting is that you can look at the biochemical values or data of the RAS nanocluster, and you tend to find that this is the data for which it matches quite well. So which means that RAS nanoclusters tend to be desorption dominated. And this is interesting because now we can take this and compare with the data that we had before, but unfortunately our model prediction doesn't really match with observation. So our observation was that there was a power law followed by an exponential decay. Instead, we find that it is kind of like an exponential decay. There is no power law involved over here. Sure. So how do we explain this discrepancy? So one of the things that we missed and we kind of deliberately assumed was that the actin cytoskeleton or the actin asters are identical across all the membrane. And that is not really true. So when you look at some of the realistic pictures of the actin asters, they can be quite heterogeneous. And in fact, if you do some look at the measurements, you find that the actin aster sizes are exponentially distributed. What that means is that concentration of actin, which is proportional to the size, should also be exponentially distributed. And that means that the on rate should also be exponentially distributed. Now the question is, now that we know that the on rate should be exponentially distributed, does that explain the power law? To understand that, I'm going to go back about 30 years and look at this paper by Bouchot and Monthos, who proposed that if you have random distribution of energy barrier, it can actually give you power law even if you have exponential distribution of lifetime. So what they showed was that suppose you have a trap of some depth E, then the average time to escape from this trap is going to be e to the power E by E1, where E1 is some constant. And if you also have the distribution of trap depth to be an exponential with some mean value of E0, then you can show that the power law, it will have trap residence time will have a power law with an exponent that is given by these two constants. So k on is analogous to E over here because we have an exponential distribution. The question is tau c is also like that? So we find that tau c is indeed exponential on k on, and when we compare this with that, it implies that we should have a power law that is of this particular form. And when we compared with the exponential data, our data, it matched exactly with the short times. So we basically said, okay, it is at least matching the short time prediction. So can we take this value and put it into our model and then predict the actual distribution? And voila, we find an exact match with the experiment without any sort of manipulation of the parameters, okay? And what is interesting is that this 20 per second is exactly the rate at which KRAS GTP is hydrolyzed to KRAS GTP. And it is well known that KRAS GTP forms cluster, KRAS GTP does not form cluster. And that basically gave us a confirmation that cluster lifetime is the key determinant of the residence time. So with that, I'd like to thank Debanjan, my funding agency, and this is the paper that we just published, and I'll be happy to take any questions. Thank you. Yeah.
Are you not taking the clustering dynamics of the actins, or is it?
I'm not taking the clustering. I'm assuming that the aster is always going to be there. The aster dynamics is so fast that there is always going to be an aster. Then the protein dynamics, protein kinetics is what is ultimately determining the, yeah.
Hey, Sumantra, nice talk. So have you tried non-constant off rates? Because it's very well known that off rates, the unbinding rates are not constant. They depend on the forces between these actin polymerase. And so if you want to take a off rate that is not constant, would you still be able to explain these results?
I did not know that, so we can definitely try, but we just wanted to start with a parsimonious model that will explain our data. And this seems to explain the data quite well, even for a broad range of values. So we stuck with that, but that is something we can definitely
But
continue.
is there experimental measurements that show that this k-on is distributed exponentially?
Well, experimental measurements does not show that k-on is distributed exponentially. What it shows is that the aster size is distributed exponentially. And because the concentration of actin is proportional to the size, and we assume that the on rate is proportional to the concentration of actin, so from that, we basically get that the on rate is exponentially distributed.
Okay, let us thank the speaker.